What's up, guys? Welcome back. We're going to continue with the PowerPoint on the reproductive system. Specifically, today we're going to focus on fertilization, gestation, a lot of crazy words. We're going to truck through it. It's going to be a good one. Um, if you recall from the previous lecture, we talked about specifically the male and female reproductive system, the structures, more importantly, spermatogenesis and oogenesis, and how we form these gametes. We recall that these gametes are haploid and have half the number of chromosomes, and thus the egg, aka secondary oocyte, and sperm will have to fuse to form the zygote. That's pretty much where we pick up. Um, Good video. Check it out. Summarizes everything. This was from the previous lecture. This is where we're getting to now. Then we're going to go through into implantation, embryonic and fetal development, and then briefly at the end, we will talk about birth. So these are the steps, right? Assuming that we had ovulation, we have fertilization. Maybe we don't, but then we wouldn't be talking about this. So we're going to assume that fertilization has occurred, right? Obviously, fertilization usually happens within the uterine tube or fallopian tube, approximately half a day to a day post ovulation, right? So those sperm can be in there for a little bit ahead of time, usually have kind of like a 3-ish day window. And then once that zygote forms, we have cleavage. We'll talk about that. It turns into a morula. It turns into a blastocyst. It implants, right? So obviously all of this is going to be taking place, not all of this. The implantation will be taking place in the endometrium. So step number one to go through this, get the big picture, the big roadmap, and the steps are fertilization, cleavage, morula, blastocyst, implantation. If you can kind of remember these steps, each one of them has a multitude of things we need to talk about. But you could see here as well that there's a time frame associated with this growth. So fertilization, usually 12 to 24 hours, right? Cleavage, usually completed about 30 hours post-fertilization. We'll see that morula in about 3 to 4 days. We'll see the blastocyst in about 5 days post-fertilization. And implantation usually occurs about 6 days to a week post-fertilization, right? Let's dive into it. Fertilization, right? We have a lot of sperm that get released via the semen, which we talked about. Only about 0.0001% of the 200 million sperms will make it to that oocyte, right? So they got to go up through here. They got to go boom, right? So 200 million released, 2 million, right? Make it to the uterus. 200, very low percentage, may potentially reach that secondary oocyte, right? So the more sperm, the higher chance some of those get there. And what happens? So in order for the sperm to be more or less functional, we have a process called capacitation. And this is basically your sperm becoming active, if you will. So fertilization cannot occur until this process is complete, right? In the epididymis, we talked about the kind of that maturation, but the sperm has to gain competence and hypermobility, right? This happens in the female reproductive tract, right? So one, it has to be able to swim super, super fast, and it has to have the ability to bind to the secondary oocyte, right? So you can see this is all covered with cholesterol and whatever. It's mature, right? It looks like a sperm, but it's not fully functional as far as fertilization capabilities go. A key thing here is we will see the acrosome, right? The acrosome will play a very, very important role once this sperm attaches and has to kind of dig through the outer layers, if you will, of that secondary oocyte. So capacitation is very, very important for functional fertilization capability. Makes sense. The secondary oocyte's actually pretty big compared to, respectively, other cells in the body. So you have this 3-day window I discussed previously for fertilization. Basically 2 days before ovulation, because that sperm takes kind of a while to get there. They're not the smartest creatures. They follow a chemical gradient, so they know which side to go in, obviously, because it could be in either of the fallopian tubes. And some of these chemicals are basically calling that sperm towards it. And obviously, you cannot have fertilization unless there is ovulation. So it kind of buys us a little bit more time because those sperm take a while, right? They could kind of get there and then ovulation occurs if kind of moving towards there in the process. So makes sense. If we recall, when we looked at these pictures previously, part of that mature or Graafian follicle, this oocyte had two components that were mentioned. The zona pellucida and the corona radiata, right? So once this oocyte is released, we see that it comes with, obviously during release, it's now a secondary oocyte, or during ovulation, I should say. It comes with both of those components on it, right? The zona pellucida and the corona radiata, and it's important, right? So that's how it's ovulated, and then it looks like this, right? So the layers of those secondary of the, I should say, surrounding the secondary oocyte, sperm's got to get through, right? It's got to get to the middle, right? So it has to go through the corona radiata, which is the outer layer. Think of it like like rays, if you will, kind of off of that oocyte. And the zona pellucida, which is made of glycoproteins and things. So it's going to have to find its way through there, right? Think of it kind of like as a protective coat. So what we have is we have the acrosome kind of at the head of the sperm. And that's going to be the first thing usually to make contact, right? And the acrosome contains enzymes that are proteolytic. So basically what these are digging enzymes. So in that acrosome, once the sperm hits, right, these enzymes start to more or less dig its way through these layers of the corona radiata and the inner zona pellucida, right? So that is called the acrosome reaction. It is important so the sperm can get inside and deliver its genetic or nuclear material, right? And recall, this sperm is a gamete, so it is haploid. It has 23 chromosomes. So the first sperm to attach, right? There's a receptor called the ZP3 receptor, whatever. It's chilling there. It looks like these dots. Next picture will show you something slightly different. And kind of as that sperm digs through, it binds to this receptor, right? First sperm there, super important. We have that adherence of the receptor, right? Post-acrosome reaction, and you can see that it fuses. And as it fuses to that receptor, it can inject, if you will, that nuclear material into the cytoplasm of the oocyte, right? So acrosome reaction, important, right? Capacitation, important. Acrosome reaction, important. And then fusion with that ZP3 receptor to allow adherence, and then entrance of that nuclear material into the cytoplasm. And this shows a pretty cool picture of the sperm in contact with that secondary oocyte. And after that sperm fuses with the membrane and the nuclear material is present in the cytoplasm, um, we want to make sure that no other sperm can get in, right? When you have multiple sperm getting in, that's called polyspermy. Not good, right? We want one nucleus fusing with one nucleus. So what happens after that acrosomal reaction, right? And I like this picture because you can kind of see what those receptors look like there. Sperm binding receptors, ZP3s. Once this enters, I should say, kind of post-acrosome reaction, the oocyte gets charged, right? Think of it like a kind of like a positive charge that flies through this thing, and it helps block polyspermy. In addition, the receptors get destroyed, right? So we have that shocking, which is a fast block of polyspermy, and we'll see a chart that's crazy, but I'll show it to you anyway. And then you have the degradation of those receptors, so no other sperm can bind, right? So these are two things that happen, and that's obviously a slower process. It's going to take a while to kind of break down and degrade all those receptors. That's why it's called a fast and a slow. And this is what's actually going on, right? So sperm binds, blah, 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 positive charge, right? Fast block of polyspermy, right? All this stuff happens, leading to the receptors being degraded. And we have a slow block of polyspermy. You don't have to stress about all the specifics, but you should understand that both of those are occurring to prevent polyspermy. We talked about meiosis 1 and meiosis 2 in the reproductive system when I showed you this picture, kind of about the development of the oocyte to the primary oocyte to the secondary oocyte. So where we're at here is this part, right? Everything else was ovulation. And kind of along with that follicular development, we talked about all this, right? So here, meiosis 1 completed before ovulation, but meiosis 2 is completed after fertilization, right? So this is what got ovulated. Here we have fertilization, right? We'll get rid of that other polar body, and that ovum fuses with the sperm and completes meiosis 2, right? What this looks like is gamete fusion, if you will. The sperm nucleus and the ovum nucleus fuse together to form one single cell nucleus. So you got the 23 here, you got the 23 here, and then this is called syngamy. And that's when you have your one cell that has 46, right? So a couple processes involved, some protective mechanisms to prevent polyspermy, a little bit of protection around the oocyte. Now this cell is going to divide into two cells, right? We call the original syngamy, we call that a pronuclei. But this cell is going to turn into two more cells, which is going to turn into two more cells. So what you get is you get this mass of cells growing. And that's pretty much where we're going to continue with throughout the lecture, right? We have this zygote. That is the first cell that I showed you on the previous slide after formation, after the syngamy, that pronuclei, post-fertilization, we got it. First cell. What happens to this cell? This is done. Boom. We got it. Fertilization. Next thing we're going to have is cleavage, right? So what does this look like, right? It's going to turn from that phase to a morula to a blastocyst, and it gets a little bit more complicated as we move through this. So we have zygote cleavage, right? It's kind of like that, not the best picture I drew before, but one cell turns to two, two cells turn to four, blah, 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 right? So that zygote is the two-cell stage, right? Or I should say, once that one cell divides via cleavage, right? The cleavage is basically the continued splitting of these cells. Four-cell stage, morula, blastocyst, same picture as this, just showing it from an internal view if you kind of cut it open. So we see the increase in cell number, right? Still within that zona pellucida, but the increase in cell number as we progress through from day 1 to day 5. Depending on the amount of cells and the phase that it's in, we call it something different. So we have a 2-cell zygote, 4-cell zygote, a morula, right? Lots of cells in there. And the blastocyst actually starts to look like something, right? Just looks like a cell mass, but it looks like something. So to bring it back to the female reproductive system, we had ovulation, fertilization occurred. That's not a very good sperm, right? Met up with that secondary oocyte. Meiosis 2 was completed. What do we still have around, right? We have high levels of progesterone, and we have that corpus luteum. We also have a uterus that is in the secretory phase and well prepared to receive the developing embryo, right? So, what the heck is the blastocyst? Right? That's where we're at. We had the fertilization occur. Yay. What we have is a group of cells. This will become the embryo. These cells are called totipotent stem cells. This means that they can turn into a bunch of different types of specialized stem cells. What a stem cell is a cell that can differentiate or turn into different types of cells. These cells can literally pretty much turn into anything, right? And they will, as we see this development occur, start to specialize, right? So the embryoblast will be the embryo, right? The trophoblast, which are these cells towards the outside, will become the chorionic sac and the fetal portion of the placenta. Right? We have the blastocele, which is basically that fluid in there, and that zona pellucida is still chilling as kind of the outermost layer. Right? So these embryonic, the embryoblast or these embryonic cells will start to turn into, right, more specialized cells. So what happens next? The blastocyst, if you will, hatches out of that zona pellucida. Up to this point, we kind of had that protective coating on the outside, and the blastocyst will implant, right? So this is your zona pellucida. That blastocyst sneaks out of there. Clearly, this is the embryoblast here. You can see the trophoblast cells, and that blastocyst leaves. What happens now? Implantation. Implantation is when the blastocyst adheres to the endometrium of the uterus. This is what we call implantation. It has implanted itself into the endometrium, and obviously that endometrium is in the secretory phase, right? It's got all these endometrial glands. It's well prepared to receive such a blastocyst. So it adheres. This kind of shows you I don't know why that keeps happening, a picture of the adherence of that blastocyst. You can see that this embryoblast, kind of this face, attaches to that endometrial wall, right? We can still see the trophoblast, but you notice That protective layer is gone, and that embryoblast is going to start doing some things, right? Before it can start really developing, it's going to have to get itself in here so it's attached and safe. Um, and we will see how that happens, right? It burrows deeper, if you will. The trophoblast, let's see, specializes, right, into two layers. So that trophoblast, that outermost layer, if you will, um, turns into this thing, right? A cytotrophoblast. And this is important. Not so much the name, of course, maybe I could ask you a question on it, but what's important is this specialized cell, these specialized cells produce HCG, human chorionic gonadotropin, usually around day 8. This HCG does very important things. It tells the corpus luteum to stick around. We can test for it. Um, it is very important, right? But it's only produced at this stage. And the syncytotrophoblast um, has digging enzymes and things, proteolytic enzymes that allow that to kind of sneak in further into the endometrium and get safer, if you will. So the presence of that HCG is important, as well as that blastocyst kind of working itself into the endometrium. And like I said, if fertilization occurs, this was from the previous lecture, HCG is present. It tells the corpus luteum to stick around, stay alive, and then it starts producing various hormones, right? Clearly, we talked about the maintenance of that endometrium as well, which is creating a favorable environment for the implantation of said blastocysts. This is what the implanted embryo looks like, right? It kind of goes in there, kind of has a capsularis, kind of a protective layer. Now it is inside the endometrium, right? We have the pseudobasalis, right? It starts to look like something, and we'll kind of flip back and forth. You can see some of these cells in here are starting to reorganize themselves. We're developing the yolk sac and things that we will see as we move forward. Now it's in there. Cool. What happens next? We have embryonic and fetal development. This is when it gets kind of crazy, right? The last one was crazy too. Obviously, you could check out the video. Super, super important. Kind of just as a different way to look at what we're discussing. So, in theory, post-fertilization, 9th day-ish, a little bit over a week, the blastocyst should be fully implanted. This is the picture I showed you before, right? Now, a few things happen. We can see this guy, which we'll talk about in a minute, right? We can see those trophoblast cells. Here we have the presence of a yolk sac. It's always kind of yellowish on the pictures, like egg yolk. It supplies nutrients, source of blood cells. Basically, it's like a food factory kind of temporarily. And then we have the formation of these lacuna. These little spaces will be filled with maternal blood, right? We can see the syncytotrophoblast there that helped dig it in. Because the blood flow will be super important for the development of these cells, they need nutrients. That lacuna formation is pretty significant, right? And you can see that there's a little space here called the amniotic cavity, right? The amnion's this layer right there. And the amniotic cavity will turn into the amniotic fluid. And the amniotic fluid is what's going to be surrounding that fetus through development. It acts as a shock absorber. It helps thermoregulate, maintain temperature. Water is a very good conductor of heat. And it obviously keeps the fetus moist so things can grow and it can be safe. As we progress, right, We have these extra embryonic mesodermal cells that formed. These weren't here before, right? Acts kind of as connective tissue. Um, and you can see, let me see here, um, the term chorion, right? So we will mention as we go through, I don't know, a bunch of slides, chorionic villi sampling, right? You can be familiar with that. But the chorion is basically the extra embryonic mesoderm, these red cells right here, the cytotrophoblast and the syncytotrophoblast that we talked about before, right? This will form the chorion. We can take samples of that, run analyses, get some idea pretty early on as far as genetic issues and things, because those cells are the same cells that originated from the first zygote. So they all have the same genetics. Um, as we go through, we just see a little bit more evolution, if you will, right? We can see that extra embryonic mesoderm, those red cells kind of fully surround it, right? We can see that amniotic cavities slightly enlarging. There's just more cells going on. We can see the yolk sac, right, which we talked about before. And now we have the presence of the connecting stalk, right? You can see that this is kind of separating out um, off that connecting stalk. This will be important, right? Now we start to have the maternal blood flow. Come on, maternal blood flow. Start mixing in with those lacuna, right? We start to see something that looks very much like an umbilical cord. We see the amniotic cavity really start to take shape. And we see the embryo start to look like something more than just a mass of cells, right? It has these little dots in it. It's starting to take shape. Obviously, it's still connected to the yolk sac. And this is what it looks like at about 3 weeks post-fertilization, about 21 days. It seems simple, but there's a lot of cellular activity that's been going on to kind of establish all of this properly, right? Here you have, and it's just kind of jumping ahead a little bit. You can see the umbilical cord. That's a horrible color. You can see the umbilical cord. You can still see the presence of that yolk sac, but proportionally it's much, much smaller. And here you can see, let's see if I got a good picture, that chorion that we mentioned. Here you can see the amnion and you can see the amniotic fluid, right? We'll mention as we progress through amniocentesis versus that chorionic villi sampling. And it's just showing you that amniotic cavity or the amniotic fluid in there, as well as some of the development of the umbilical cord, but we'll get to there. This is showing you the, oh man, the germinal, embryonic, and fetal stages. The germinal is the first one, usually first 2 weeks, 0 from fertilization on. Basically, what you have during this germinal phase is a cluster of cells. Then you have the embryonic phase, right? Usually up to 8 weeks post-germinal phase. And you start to have some of those cells start to differentiate. You can see 3 major areas of the brain. It starts to look like it's got some limbs and things. And then 9 weeks until birth, we refer to it as the fetal period, right? Obviously, neural tissues developed, limbs, et cetera, et cetera. And we'll kind of take a look at what that looks like. But those are the 3 general phases of which we have covered that pre-embryonic or germinal phase. What I like about this picture is it kind of simplifies what the heck was going on in case you got lost along the way. And now you have your embryo, right? We'll start to have some organ formation and things like that. as you progress through post week 9 until birth, right? 38, 40 weeks-ish. It is now called a fetus. And it primarily grows in size, and some of its organ systems really start to finalize development. So, what happens here? This is the initiation, if you will, of that embryonic period. The key thing, come on, is we start to see, and I think we see it on the next slide, the formation of 3 germ layers. And I'm going to tell you what that is, right? We start to see at that 4 to 8 week period, we start to see organ development, right? You can see eye, ear buds. You can see limb formation a little bit. So we start to see, it's just going back to the other one, right? The epiblast, we talked about that. The formation of the 3 layers. And these 3 layers, right, coming off that, oh God, coming off that epiblast, if you will, and hypoblast region, we see the endoderm, mesoderm, and ectoderm formation. These are the 3 germinal layers that we're talking about, right? This is referred to as gastrulation. And those 3 layers are specialized stem cells that will turn into all the stuff that we need, right? So not to be confused with endometrium and stuff like that. The dermal are your 3 layers, ectoderm, mesoderm, and endoderm. And we can see that these cells start specializing. And it says here they differentiate, right, off that primitive stalk that we talked about. This is what they look like, right? Ectoderm, mesoderm, and endoderm. And this is what they turn into. Should know this. Most of this was briefly mentioned in A&P1 when you talked about the development of each of the tissues, right? Muscle, bone, all that stuff. You hopefully recall hearing about ectodermal cells turning into the skin, ectodermal cells turning into the nervous system, mesodermal cells turning into muscle, connective tissue, and bone, endodermal cells, more internal organs, GI, digestive, respiratory tract, as well as a bunch of other organs. So these dermal cells are very important in developing the actual organ structures inside. This, right, we talked about nervous tissue, epidermis of the skin. You should have a general idea. You don't have to go through and memorize this entire chart, right? We talked about GI tract, internal organs, bladder, things like that. Glands, right? Endocrine glands we discussed. The mesoderm is going to be muscle tissue, cartilage, bone, obviously, respectively, bone marrow, things like that, right? So that's what these 3 layers will differentiate into. And that's important, right? We have neurulation, which is basically formation during this period, right? Again, embryonic development, 3rd week-ish. We form a neural tube, right? The neural tube, right? This is just that cluster of cells, right? That was found kind of by that embryoblast, the epiblast and the hypoblast. We have that neural tube that will differentiate into the spinal cord and brain, right? There are a variety of different issues that can occur if that process that we just mentioned does not happen perfectly, right? So this just gives you a list. It kind of describes, you have things like spina bifida, anencephaly. We'll see a couple pictures, but that's the significance of that development of that neural tube properly, right? Could be missing part of the head. You could have your spinal cord coming out through your vertebral column, things like that, right? So we see here spina bifida and anencephaly. One thing that's important to that notochord formation is something called folic acid. So this is more of an informative thing than necessarily something I'm going to ask you a ridiculous question on. Um, but folic acid is important, right? There are some issues where people kind of can't metabolize it properly, but the folate helps with DNA and RNA production, protein metabolism, and there's a lot of that going on during development. Um, and it's really, really important for periods of rapid growth, like this period we're talking about right here, during pregnancy and fetal development, right? There's other ways you can get it without supplementation, eating good foods, dark leafy greens, etc. So at this stage, we have the formation, right? We can see that neural tube that turned into the neural plate. We have basically the formation of a base level heart, right? Within this cardiogenic area, we will have a very simple heart form, right? There's endocardial cells. They kind of fuse together. They make a heart tube, if you will. And you have what is, in theory, a functioning, very simple, basic heart. You have base level atrium, base level ventricles, right? Base level veins, right? The sinus venosus. Um, and although we have circulation of blood, you can't really detect a heartbeat till a little bit later, right? Usually 6 to 7 weeks-ish, 2.5 months or so. I'm sorry, I should say almost 1 month. No, 2 months. How many weeks are in a month? 6 to 7 weeks, a little bit less than 2 months, right? You will be able to detect that heart rate usually. Um, we have other organogenesis, right? Um, other organs will be formed, right? Some of that tissue specializes, starts to grow via its pre-programmed kind of genetic establishment. Um, and you start to have clear human-like characteristics at the end of 2 months. Granted, the thing's pretty small. Um, the embryo gets big rapidly, more or less, because it was really, really small. And now it's actually more or less visible. And at the end of your second month, right, um, it starts to take on those characteristics, right? We saw this picture, like the 4 week embryo had some things formed. And basically, we're going to go through the slides from week 4 to week 6, if it lets me click on this, to week 7, right? And we talked about what was being developed during these phases, but you could definitely see it start to take on more characteristics, and that's what I was showing you. At the end of the second month, right, that 7-ish to 8 weeks, um, you really have established human-like characteristics, right? There's some bones, that heart's formed. We have limbs, shaped head, ears, etc. Right? That's what it looks like. Clearly, it's not very big. Um, that's an 8 week embryo. Now, there are some critical periods, and this kind of summarizes it. Clearly, if you had issues over here, um, with implantation and any of that gastrulation, um, you're going to lose it, right? Um, as this progresses forward, you'll see kind of the heart, which we talked about was one of the first the first established organ, which is important to circulate blood. Then you have your limb formation, right? And these are critical periods. So although some of it starts to form early, right? Like in the beginning, you have like the CNS, you saw like the three primitive stages of the brain and the heart. These are critical periods for development, right? And you'll see that some of these major ones are around that 6 to 8 week period. Um, and things later on, eyes, mouth, palate, talk about cleft lip, genitalia, ears, and CNS will all develop a little bit later on. Keep in mind this is scaled, right? This is 8 weeks, and this little bit right here is 9 to 38, right? That's fetal development. So we talked about embryonic development and fetal development, right? Thus, embryonic checked off. The last one is the fetal development period. It lasts the longest, primarily. Obviously, it's called the fetus now. It grows longer, kind of continues that organ development, and more or less is independent as far as its growth, right? All it needs is circulation of nutrients from the mother. And how does it get that? It gets that through the umbilical cord, right? So you have a placenta, right? And you have the umbilical cord that's feeding this placenta nutrients. What's different here is you have 3 vessels. You have 1 vein and 2 arteries. And just like in the respiratory system, those switch. So the vein is actually carrying oxygenated blood, and the 2 arteries are carrying deoxygenated blood. So the placenta is kind of like the respiratory system in which those arteries and veins switch colors. Makes sense, right? This is highlighting the fetal stages and what kind of goes on, right? So you have the 9-week, right? Which we just showed kind of, you could start to see some specialized features and stuff like that, right? Sex organs differentiate, finger and toes develop, hearing begins, lungs begin to develop, brain grows rapidly, bones fully developed, muscles fully developed. Thing is huge. And you can see that that growth increases rapidly over this time. And you have a video here, you can totally check that out. And this talks about some changes and weight during embryonic and fetal development, right? This specifically showing you the fetal. Here you can see heartbeat detected, right? Focus on the things that are underlined. We're going to talk about this vernix caseosa. That's important. We're going to talk about lanugo. That's basically like light hair. That vernix was kind of fatty secretion kind of coat around the baby. At this point, about 20 weeks usually, you have movements that can be felt, right? Muscles are developed, things kicking around. Here, around that 20 to 25 week period, about 6 months, they can survive if born prematurely, right? They might have issues and stuff like that. But that's the kind of the point at which the majority of development has already taken place. This talks about testes, bone marrow, etc. At 30 to 34 weeks, we have the ideally upside down position taking place in preparation for exit, right? Obviously, you have breech babies that could be transverse breech, they could be right side up. But basically, the idea is that fetus will assume that position preparing for birth, right? Cool. This was the vernix, right? Upon obviously labor, and this is the lanugo. So, ever seen it? You know exactly what it is. And each of these serve their purpose as far as kind of insulating the baby, etc. So we actually talked about this in A&P1. In fetal circulation, the blood doesn't have to go to the lungs per se. So we have many structures, right, that kind of reroute blood from, let's say, maybe the right side of the heart to systemic circulation. One of those is called the ductus arteriosus. We have the foramen ovale, and all these were mentioned in the heart and AP1. We have some things that are involved with liver. Obviously, here you can see the umbilical cord, veins, and arteries, respectively. But specifically in relation to that heart, there's the ductus arteriosus and the foramen ovale. The foramen ovale should close up, obviously, and it becomes the fossa ovalis. But what that allows for, even though I just kind of scribbled on here, that allows blood flow to move from the right atrium to the left atrium, bypassing pulmonary circulation. Even if blood does get pumped out through, which it will, the right ventricle, it goes up. We have the ductus arteriosus, which connects that pulmonary artery directly to the aorta. So an additional way we can kind of bypass lung circulation because the baby's in liquid in the amniotic fluid and it's not breathing air. Thus, the amount of pulmonary circulation it needs is decreased, right? Cool. Check, check, check. And this just describes that. That foramen ovale, right here, and the pulmonary trunk to the aorta, the ductus arteriosus. So basically two alternate flows of blood to kind of bypass, if you will, the lungs. We mentioned both amniotic fluid testing and chorionic villi testing. We will make note that chorionic villi can be done earlier. Sampling of some of those cells to do genetic analyses on an amniotic fluid because you need to wait till you have a little bit more fluid in there is usually done a little bit later. We call that amniocentesis and CVS, chorionic villi sampling. Right, this is the position of the organs. You can see obviously during that uterine expansion, baby's getting bigger, things get kind of pushed out of the way. Digestive system moves up, bladder's going to be compressed, ovaries kind of move off to the side. You can see them over here. And what happens in the mother? A variety of different changes, right? The mother's pregnant. We talked about the baby the whole time. The mother's the one that's doing all the work, right? Granted, the baby's grown. It's doing a lot of work too. But because there's so much increase in metabolic activity in the mother, we have increased in heart activity, right? Cardiac output and blood volume go up significantly, right? There's more tissue that needs to have circulation of blood. We need more oxygen, right? So resting volumes of breathing go up, right? We need more air. We need more oxygen. There's more activity. This oxygen needs to be circulated. Kidney activity is going to increase, right? Glomerular filtration rate increases. We'll talk about that in probably the next lecture. When we go through the kidney, we'll talk about what GFR is. More frequent urination. In addition, that bladder is smaller, right? Because it's being compressed by the uterus. And GI activity goes down. The demand for nutrients goes up. But we want to make sure we're maximizing all that nutrient absorption, which we will talk about in the last section in lecture. So these are all physiologic changes that are happening in the mother's body, right? Because of this, here we can see the positioning. We can see that placenta. We can see the umbilical cord. We can see the head down position, and we can see the presence of that cervix and vagina. And you can almost start to see the trajectory that this baby will take, right? Obviously, it's in the amniotic sac right now, filled with amniotic fluid, just hanging out. So what needs to happen, right? In order for this baby to come out is cervical dilation, rupturing of the amniotic sac, right? That will open up the cavity, allowing for the head of the fetus to exit. And the rupturing of that sac frees up so it can leave, right? The placenta is located right here. The baby's head obviously comes out first. Clearly, this was your cervix with extreme dilation, and they'll be like cervical dilation is XYZ centimeters. And that's how far along this process is. We'll talk about the positive feedback associated with parturition. We call this stage expulsion, right? We call this stage dilation. And here we have, and we talked about this with the endocrine system, an oxytocin positive feedback loop. Now, this causes uterine contraction. We have to squeeze the uterus to push against the pressure of the opposing cervix, right? And things. So what happens is the cervix starts to stretch. That stretching sends signals to the brain, tells the pituitary gland to release oxytocin. Oxytocin, like all other hormones, enters the blood, right? Goes to the uterus, and oxytocin stimulates uterine contractions, pushing the baby out. What does that do? Stretch the cervix. Let do circle. That stretching of the cervix sends nerve impulses to the brain, where we release more oxytocin, causes further uterine contraction. So instead of something kind of counteracting, positive feedback is promoting. So the oxytocin triggers contraction, which promotes stretching, which triggers oxytocin, which promotes contraction, which triggers stretching. More oxytocin, right, leads to expulsion. Baby's out. Everything's good. Now we have the placental stage. So that placenta that was in there, that was kind of the bridge between the fetus and the mother blood, super important, is expulsed as well, right? So we call that the afterbirth. The umbilical cord as well as the placenta will be expulsed as well. And these are general gestational breakdowns as far as early term, full term, late term, post term, in relation to that 37 to 42 week period. This has some other included information videos you can check out as far as episiotomy. Obviously, the baby's head's coming out, right? The space is limited. Sometimes they have to make incisions to free up a little bit more space, as well as this talks about epidurals and what the role is with that as far as pain management and some other things that they would give you to help promote uterine contraction and increase the effectiveness, if you will, of the labor. This talks about lactation. We actually mentioned this when we did hormones, but you can totally check it out as far as the mechanisms of prolactin is involved in milk production. Oxytocin is involved in milk ejection and the suckling effect. That has an influence on the hypothalamus as far as breastfeeding goes. And we made it. So in this section, we literally talked about all of the steps, phases, terms, and anatomy from fertilization until childbirth. You got to see how things developed. I tried to emphasize the important terms. But other than that, I will see you in the next one. Study hard.